Good evening, everyone. It is my great pleasure to welcome all the participants, faculty members, researchers, professionals, and students to today's session of the 21 Days International Summer School Training Program on Climate Resilient Agriculture and Allied Sciences, organized by Edu-Mentors Pvt. Today, we are privileged to have with us Dr. Anju Pathania as eminent academician and researcher in the field of plant breeding. Dr. Pathania is currently serving as an associate professor of plant breeding and is also the head of the Faculty of Agriculture Sciences at DAV University, Jalandhar. She has more than 15 years of experience in plant breeding, molecular breeding, disease resistance breeding, and crop improvement with a strong focus on developing improved crop varieties for sustainable agriculture. She obtained her PhD in agriculture plant breeding from CSK Himachal Pradesh Krishi Vishwavidyalayam, Palampur, and has also qualified the ICAR and ET in both plant breeding and genetics. Throughout her distinguished career, Dr. Pathania has worked with prestigious organizations including DAV University, CSK Himachal Pradesh Agriculture University, and Sangeeta Private Limited. She has successfully led and contributed to several research projects funded by organizations such as the Department of Science and Technology and the Department of Biotechnology and CSIR, focusing on crop improvement, molecular breeding, and disease resistance. Her academic contributions are remarkable. She has published numerous research papers in reputed national and international journals, authored several book chapters with renowned publishers, guided MSc and PhD scholars in agriculture and research, organized national and international seminars, workshops, conferences, and educational programs. Dr. Pathania's expertise in agriculture, in plant breeding genetics, biotechnology, and sustainable crop improvement has made her a respected name in agriculture science. We are truly honored to have such an accomplished scientist with us today. I am confident that her lecture and will provide valuable knowledge and practical insights to all participants. Without taking much more of your valuable time, I respectfully invite Dr. Anju Pathania to deliver today's lecture. Ma'am, the virtual platform is all yours and welcome and thank you for joining us.
Okay, thank you. Am I audible? Hello, am I audible?
Yes, ma'am.
Okay, and the presentation is visible.
Yes, ma'am.
Okay, so we will start our lecture. So, good evening, everyone. So, today we are going to discuss or learn about integrating genome editing and high throughput phenotyping for climate resilient crops. So, this topic lies in the heart of plant breeding, molecular breeding, biotechnology, artificial intelligence, and various multi-omic techniques. So, why this topic is important? Because the climate change is the hot topic or hot area of discussion as well as research in today's time, and it has resulted various challenges in the field of agriculture, affecting the productivity of the crops, resulting into various changes in weather parameters like temperatures, increasing temperature. There are frequent droughts, problem of salinity, then there is frequent floods, erratic rainfalls, and with the change in climate or global warming, which occurs due to various reasons, due to industrial processes, deforestation, and other anthropogenic activities, resulted into the emergence of various pests and diseases, which ultimately affect the crop. So these changes also affect the crop growth, its flowering behavior, and the fertility of pollen grains, and even the finally the yield of the crops. So climate change has also affected the various other parameters like we need feed, fiber, or other bioenergy fuels for the rapid growing population. So all these are affected due to the changes in the climate. At the same time, there is continuous increase in the global population, and the requirement of food grain is increasingly continuously increasing, but the area for cultivation is continuously shrinking. That we all know, that the population, it is predicted that by 2050 the global population, it will reach to the mark of 10 billion people. So for this continuous increasing population, again we need food. And to ensure food, the conventional breeding approaches which we are using are not sufficient, and they are not at the pace with the changing climate. So we need to integrate various molecular techniques, including genome editing, high throughput phenotyping, to enhance the or to increase the process of developing new varieties. And in this case, we need to develop varieties which can tolerate the changing climate scenarios. Like we need varieties which can tolerate the water deficient situations, which can tolerate the flooding conditions. And these crops, they have to adopt to these changes, or they have to be resistant for these stresses in single growing season. So I mean to say that the crops, they are experiencing multiple stresses due to these changes, climate changes, in a single growing season. So we need to develop varieties which are resistant to drought also, which are resistant to flooding, frequent flooding also, and which are tolerant to heat stress also. And at the same time, they should be efficient in utilizing nutrient. Means we need to put minimum nutrients, fertilizers, so that the production costs reduce. Then we need, since water scarcity is there, so we need crops which use less water and produce more food grain. So again, we need varieties which are resistant to insects, pests, various insect pests and diseases, which whose incidence also increases with increase in the temperature. So the stability of the yield also influenced under variable environmental conditions. So the genotypes which perform well in one environment, they can't perform well every time in another environment also. So we need varieties which can perform better under different environmental situations, and which has also nutritional quality. Means at the same time, yield should be there, resistance should be there, and nutrient content should also be there, to avoid the malnutrition or to attain the nutritional security. So means in one variety we need so many traits to be introgress, which are very very difficult to introgress through conventional breeding approaches, because these traits, they are controlled by many genes, and it is difficult to control these traits using conventional breeding approaches. The conventional breeding approaches simply involves of developing superior plants, and for that purpose, means what we do in to develop new genotypes or new varieties or improved varieties, we just create variations, and we try to select the desirable plants. So to create variation, there should be traits in the population or in the germ plasm, so that we can utilize those traits either through crossing, hybridization, or mutation breeding. So using different techniques to develop the improved varieties. And first we create variation, and then we select the superior plants. So that is we do in every breeding method. But the problem with conventional breeding method is that it took a lot of time, means 10 to 12 years to develop a variety. Okay. And then the traits, they are controlled by many genes, they have influence of environment also, so we call them as quantitative traits or polygenic traits. So the selection of such traits is very difficult through conventional breeding methods. No doubt that the marker assisted selection is there, genomic selection is there, na? And genome wide association mapping is there. So these are all molecular biological techniques which we use, but these techniques only enhance the selection efficiency of the breeders, but these techniques cannot create the variation. So to create variation rapidly, fast, with changing requirement or the objectives, we need to have some innovative technologies or techniques. Another important limitation is that we need to screen large number of population, and the characters or like such as drought tolerance or flood tolerance, whatever abiotic or biotic stresses are there, we need to screen the developed lines or advanced breeding lines under these stresses to identify the plants which are resistant to these particular traits like drought tolerance or salinity tolerance or tolerance to insect pest or diseases. And it is very difficult to create such environments under field conditions, means uniform environment under field conditions. Suppose if you want to create drought stress, so we need protected structures where we can control apply controlled irrigation, right? But we cannot control the flooding, means once rains are there we cannot control, so we need large infrastructure. But still there are limitations that we can screen only limited number of plants under the controlled conditions. So to overcome these conditions or limitations of conventional breedings, there are new technologies, advanced breeding technologies, and genome editing is one of them, which is easy and highly efficient technology in which we try to modify a specific gene at DNA level only. We need not to make any do any hybridization. We need not to do any transgene transfer, means genetic. We need not to use any genetic engineering or transgene to transfer, but we try to modify the genes at the DNA level only to change its the properties. So it is a bit fast technique, and a wide range of variant can be created in short span of time. And then we do the selection for desirable traits in the genome edited crops and genome edited variants for desirable traits, and develop the improved varieties. So why this technique is more important and nowadays, because genome editing it helps in the precise modification of the genome of or the DNA sequences which are present in the plant genome. And how this technology helps us, again here the sequencing techniques means we have sequenced the genome of different crops, so we have the sequences available with us with different crops, and we know the sequences of the genomes of different crops. So it is easy for us. What we do, we try to identify the candidate gene, means the genes which we want to modify. We have the various databases where the sequences of DNAs are of different crops are submitted or available. So we get the information of candidate genes, their sequences from there only. And then we use this gene and genome editing tools to modify those genes and to develop a new variant which is more desirable or more useful. So in genome editing, there are three systems which are used. One is CRISPR-Cas system, which is recent one, and it is again improving. Then TALENs and ZFN or zinc finger nucleases. So the CRISPR-Cas is more important because in TALENs and zinc finger nucleases, there we need complex protein engineering techniques to, if we want to modify different genes, means we every time we have to do complex changes in the guiding RNA, and to detect the sequences which are the target sequences. So the CRISPR-Cas is more convenient and more flexible technique. So in CRISPR-Cas, what happens? There will be a guide RNA, and we use endonuclease enzymes, Cas enzyme, Cas9 enzyme. Cas9 means Cas9 enzyme is most commonly used. There are other enzymes also. So what these enzymes do, we call them as molecular scissors. So they cut the DNA at a specific site, And the guide RNA, it guides the Cas9 enzyme to reach the specific DNA sequence where it guides where to cut the that particular DNA sequence. Once it cut the DNA sequence, cell natural DNA repair mechanism, it gets activated, and it do the regular means the natural DNA repairs. And during that natural repairs, there may be gene knockout, means there may be some addition or there may be some deletions of the bases, and which causes addition or deletion or substitution of some bases, that causes the switching off or the alter the sequence of gene, and which ultimately alter the function of the gene, or and that will help in the either we activated different genes, or we suppress the undesirable genes and we activated the desirable genes. So this technology, it is very precise. It make uh, you can see here in this figure that in the cell, the Cas enzyme along with the guided RNA is introduced in. It may be introduced into the cell either through agrobacterium transformation or through protoplast or particle bombardment. Any method can be used to transfer this Cas9 enzyme along with the guide RNA. So guide RNA has known sequences, and it identify the target DNA, and it tells the Cas9 protein or enzyme, this is a endonuclease enzyme, to where to cut the make a cut in the DNA sequence. So it make double strand break at the target DNA region, and then the repair mechanism get activate. There are two types of repair. One is non-homologous end joining. In non-homologous end joining, what happens that when the nicks or the cuts are created, so the DNA repair mechanism, it activated and it try to stitch the bands, and during the stitching of the bands, there sorry, strands of the DNA, there may be some deletion or addition of the nucleotides, so it changes the sequence of a particular gene, and it alter ultimately the proteins and the product of that particular gene. And second one is the homology directed repair. In homology directed repair, what happens that a known DNA sequence is inserted, and we want that where the cuts in the DNA strands are created, there that sequence with known base pairs and known sequences, that particular strand of DNA is knocked in or added into the cut end of the strands, and again the DNA is repaired. So this is the mechanism how it works, and there are many what we can say applications of this CRISPR based tools, and this idea was taken from the system from the defense mechanism of the bacteria. Bacteria use this defense system against viruses which infect the bacteria. So from there this idea came, and this CRISPR based tool, what they can do is, one is they can create random insertions or deletions or substitution of the nucleotides at the targeted DNA sequences, or it can do the base editing, means there is no need to make cut in both the strand. Instead of that, nick is created in one strand of the DNA, and there the base editor they replace the or change the one base with the another, so they cause the transition, means adenine to adenine or cytine to, so base transition is caused by the base editing. So, and another one is prime editor. So in prime editor, again it is the editing of the bases. So it is user defined, means we can change any base with any another base in the prime editing. So then when this caste system is changes, or it create mutations or changes in the DNA sequences, so what happened, it create variants, so it diversify the nuclear bases in the targeted DNA sequence where the change addition or deletions happens. So when it cut the DNA at the target region, so in some there is, in some places there may be addition, in some places there may be deletion, or in some places there may be what we can say substitution. So there may be diversification of nucleotide bases in the targeted window. So there may be new alleles may come generate, new variants may come out. Again, these CRISPR-Cas system, it is again used to modify the but we can say regulatory elements, or the elements which involved in the regulation, gene regulation. So they can also modify the regulatory elements of the DNA also. And they can also do the epigenetic changes, means they can change the histone proteins, they can change their structure, they can also suppress the transcription of RNA. They can also change the bases in the RNA or DNA. So there are multiple applications of this CRISPR based systems. And the multiple CRISPR trait stacking can also be done using these CRISPR based systems. So in multiple trait stacking means we can simultaneously modify the genes responsible for different traits. So that is called multiple stacking or multiple CRISPR screening system, where we do the multiple editing of multiple targets at the same time. So then de novo domestication is another, means earlier what we used to do in the when we develop improved varieties, we try to cross the wild species with the cultivated one to transfer some desirable genes for drought tolerance or for disease resistance or other likewise. So, but in de novo domestication means instead of introgressing gene through hybridization from wild species to cultivated one, instead of doing that, we can what we can do using genome editing strategy, what we can do, we try to change the, or we try to edit the genes which are not desirable from the from our point of view, means like if in wild species shattering is more frequent, but the plant is very resistant to drought or even diseases or pest, but shattering is more common, then the grain size is less. So instead of transferring disease resistance and other trait, what the scientists can trying to do is that they try to modify the genes responsible for shattering in that wild species, or they try to modify the genes which are responsible for more branching habit or for small grain size. So they try to edit those genes and they try to change them into the gene after editing to change them to the compact plant type and then non-shattering type. So that is called the de novo domestication. So that is also possible through genome editing strategy. Again, stacking of complex state, as I have already discussed in earlier slides also. So what we can do, we can because we know that these droughts, the genes which are responsible for these abiotic stresses or multiple stresses, like disease or insect pest. So many of these genes are quantitative or polygenic characters are these. So these genes, there may be hundreds or thousands of genes which control these traits. And in conventional breeding approaches, it is very difficult to transfer all those genes or to identify those genes or to select the plants with those particular genes. So instead of that, in genome editing, what the scientists they are doing, they try to edit the entire gene network, means the and the all the related different genes which are responsible that for that particular traits, they try to edit those different genes which are responsible for that particular trait. So we call that as multiplex trait stacking. So multiplex CRISPR Cas technique helps in improving these traits. Then the using genome editing technique, the varieties can be developed by modifying the genes which control the osmotic adjustment, or which control the movement of or transpiration, or closing or opening of stomata. So by modifying those genes, we can develop the varieties which can tolerate the water stress more effectively. Similarly, for heat stress also, we try to edit the genes which are responsible for providing plant tolerance to a particular stress, means the rolling of the leaf, or lowering the canopy temperature, or the absorption of more water through the what we can say robust root system. So these different traits, we are trying to modify these traits by the editing of the genes which are responsible for these particular traits. And the next generation sequencing technologies helps us in identifying those sequences which are responsible for the controlling these traits. And genome editing technology, or this CRISPR-Cas technique, use those information to edit those DNA sequences. So in disease resistance also, instead of what the scientists they are trying, they are altering the genes which are responsible for the susceptibility of which are responsible, or which make the plant susceptible to a particular disease. So they try to edit those genes, and they try to alter the functions of those genes using this technology, and to try to develop the resistant varieties for the various diseases. Similarly, to improve the yield, the editing of the genes which are responsible for more grain weight, or the genes which are responsible for more what we can say a number of grains per year. So they try to modify those genes which are related with the yield, or which directly or indirectly affecting the yield. So by modifying those genes and the yield, the scientists they are trying to improving the yield of different crops. So if we could able to modify the genes for different stresses as well as yield simultaneously using these genome editing techniques, then we could able to develop varieties which can tolerate, or which has resilience to the variable environmental conditions or erratic environmental conditions which are posed due to the climate change. So once okay the varieties, or we have done the editing or modification of the DNA sequences present in the cell of a particular plant. So we modification has been done, but now the question arises that there are hundreds or thousands of edited line plants which are we need to screen to identify the desirable variants. So traditional field screening means generally what the breeders are doing, they take observations, they measure plant height, they count number of tillers, they count means they use different methods manually, they record different parameters, and then they analyze the data, and they reach to the decision whether they are going to select that particular plant or not, or that particular trait is present in that particular plant or not, means the resistance gene is there or not, resistance is there or not. So it is a very slow process, and it is also error prone, means the way of recording observation may vary from one experiment stations to another, or from one experiment to another. So in that situation, the high throughput phenotyping play important role. So high throughput phenotyping is rapid, non-destructive, and um, what we can say rapid method to uh, phenotype the large number of plants in a short span of time. So what is actually high throughput phenotyping? So high throughput phenotyping, it involves automated data. It is a automated data collection technique in which various advanced technologies have been exploited to collect the data. And in this case, in high throughput phenotyping, the data is not recorded at, okay, we took the data observation at the germination time, then at vegetative stage, then at the flowering, or then at harvesting or maturity time. Okay, so in this, in high throughput phenotyping, what we do, we use advanced imaging technologies, we use sensors, robotics, drones, we use take the help of artificial intelligence to collect the data or to record the observations on various plant growth parameters, like these phenotyping techniques. They record or take images of the plant regularly. They take tens or thousands of images of a single plant during the growing season of a particular crop. So or they take or collect observations or data on that particular field, experimental field or plot, or plants, individual plants, many times during the growing season. So it will help in observing plant continuously, means without any human intervention, means when we are not in the field, or during day or night. And these techniques, or these tools, they keep on taking observations on the plant, how the plant is reacting to the drought or the flooding, or any kind of other stress, and other how the trait characters of the plant, how the photosynthesis happen, how the leaf is growing, or how the roots are developing in that particular plant under different environmental conditions. So the different technologies like these red, green, blue imaging. So these are the digital cameras which record which take visual observations on what we can say plant height, or foliage color, or leaf color, or any other, and it take observations, take images of those particular parameters. Thermal imaging means these two tools, they measure the temperature of the canopy. So they took observation on changes which occur on the temperature. For example, when the drought stress is there or water scarcity is there, so the stomata will close. So if the stomata will close, then the canopy temperature rises. So it will take the observation on the variation in the temperature. Then hyperspectral imaging means that the different compounds, or what we can say like molecules, or which are present, or organelles which are present in the or in the plants, or the in the leaves. So this technique based on the reflectance of light. So there are beyond visual range many wavelengths of lights, they are their images are recorded, they capture the reflectance of light, like how the photosynthetic pigments, they reflect the light, or so by that they come to know the photosynthetic efficiency, or the nitrogen content present in the plant, or likewise. Then the sensor technologies, these technologies, they assess the or they sense the variations in let's say in the moisture in the field, when the moisture is less or moisture is more. So temperature is high or temperature is less, or rainfall is there or not. So different these sensors, they monitor the weather parameters or the soil parameters, or changes in the soil, and accordingly the data is collected on various parameters. Then the different platforms have been used in high throughput phenotyping. And so we can do phenotyping in controlled environment like phytotrons or like growth chambers, where these tools, they are put on the conveyor belt, and the belt keep on moving, and they keep capture the data, 3D images, or they capture the pictures of crop canopy, and they record the observations. Then there will be field based platform, like we can use drones or tractor driven drones, or automated robotic system. We can use unmanned aerial vehicles to monitor the field or larger area, to take the observations. So this is similar. So we use different techniques or tools to take data, collect the data, or record the observations on various parameters, morphological, physiological parameters of the plants, which we want to select. So this is the illustration of all different high throughput phenotyping approaches, which can be used, or which can be integrated to get more precise results. So the data, either from the greenhouse, or from the field, or from the, what we can say, grow chamber, or phytotron, that data collected through these various techniques or tools, that is processed, and uh, analysis is done. So this huge data is analyzed through various, what we can say, tools. We call it as, we take the help of cloud servers or artificial intelligence to analyze such a huge data, because it is difficult to process thousands and millions of images in a short span of time, manually or through other methods. So we use various tools and softwares to extract the data from useful data from the data which we have collected using these tools. And then finally we try to validate this data under real field conditions. When we select the plants, we interpret the data, then we validate those results under real field conditions. So, why it is important? Because the high throughput phenotyping is, here we do the selection which is driven by the data. Means we collect the data, and we do analyze the data through various tools, softwares, and then whatever the results come, we interpret those results for the detection of, for the screening of thousands of plants in a short span of time. So there's these imaging techniques, or these high throughput phenotyping tools, they do the accurate assessment of the complex traits which are otherwise difficult to record, or we need to create such environment to record those particular traits. And we do the early detection of stress responses. Means before the visible we can see that the leaf is rolling or leaf plant is building. Before that, we can assess the signals of the stress, that stress is there, through these images, or the data which these techniques or tools collected. So this will help in improving the, or it increase the selection efficiency. It helps in improving superior genotype in more better way, in more efficiently, and in less span of time as compared to the conventional method. So introduces the breeding cycle. This analysis is non-destructive. We need means if you want to study the architecture of a plant in a drought resistance breeding. So instead of uprooting plant, we these techniques or these tools, they measure the different parameters, and on the basis of those parameters, we try to find out the which plant is have more robust root system, or which can tolerate drought better way, and which cannot. So it helps in doing the non-destructive analysis. Instead of uprooting root and then measuring different parameters, we can measure the parameter by doing root scan, 3D root scan, or other imaging techniques. So the problem with high throughput phenotyping is only that the large or huge data is collected, and it is difficult to process such a huge data. So the high investment is required in these high throughput phenotyping for doing throughput phenotyping. And the environmental conditions or field conditions may also vary from one experiment to another. So it is difficult to incorporate the existing farming practices, and there is issue of standardization. Results may vary from one experiment to another and from one lab to another lab. So here comes the role of artificial intelligence, and the whatever data is generated through high throughput phenotyping platforms, that data sets, they are analyzed with the help of intelligence tools or cloud servers or different softwares. So these helps in identifying the or these helps in predicting the performance of the crop on the basis of the data collected by these high throughput techniques, and then they predict the performance on that basis, and helps in selecting the superior plants which we have edited through genome editing technology. And it helps in improving the decision also, because these decisions are data driven decisions. So these are help in better prediction of the results. So there are different engines, or we can say tools, which we use to extract the data which collected through high throughput phenotyping. So there is one, that computer vision, which what it try to do, it try to extract the or try to differentiate the plant from the weed, or from the diseased or diseased plant, or any other off type plant which is present in the field. So we use different kinds of what we can say tools or softwares to what we can say further process the data to get better results. So when we combine this genome editing and high throughput phenotyping, so it will helps in uh, getting more precise result, or we can get or move towards the precision agriculture, or we can get the plants which are more desirable and more true to the genotype. So how we go for it is, first we identify the important genes. We know the sequences of those genes. We identify the target genes. Then with the help of CRISPR-Cas system, we try to we try to modify those genes. Either we try to suppress them, or we try to activate those genes. Then we try to regenerate the plants from those transformed cells. And then after growing those plants, we want to select the plants which have that particular desirable plant gene. So to you find out or to do the screening or to identify those genes, we use high throughput phenotyping techniques, and we use AI tools to do the analysis of that huge amount of data set. And get the analysis done and the results and the interpretation of results with the help of AI tools. And on that basis, we can identify the climate resilient varieties. So there are certain technical problems also when we use these techniques or tools. The pros and cons are everywhere. So there are chances of that when we try to modify the target gene, there are chances of that changes may occur in the off target, means the non-target DNA sequences. So the modification or the efficiency of CRISPR-Cas systems, the scientists, they are improving continuously the efficiency of these CRISPR-Cas systems. And the better versions are this base editing and prime editing. So in that way, the efforts are on the way to improve this or to reduce these off target mutations. Another important challenge is the transformation efficiency, because all the crops, they are not responding to the transformation. Means in transformation means that once we transfer, or when once we have modified the particular target DNA sequences, now we have to regenerate the plant from that particular cell. Okay, so many crops, they do not respond to transformation, so it will reduce the but we can say effectiveness of the technique. Then there are the polygenic traits, again the issue, because with genome editing we can edit multiple sequences at the same time, but again in polygenic traits we don't know exactly how many genes control a particular trait. So again there is challenge to improve or to modify all those genes simultaneously, because we can modify those genes, but we need to first identify those genes. So that is the limitation. Then phenotyping, what means it is difficult to do phenotyping, because we know that the high throughput phenotyping, it requires large investment. And another thing is that many these polygenic traits, they are very difficult to differentiate. Means there are very subtle differences between the two plants varying for a particular trait. So it is difficult to do phenotyping. So again, uh, and the phenotyping of different parameters, again we need to we can say train those models or the softwares to take the correct or analyze the or collect the right or correct data. Then big data management, again the huge data collected through high throughput phenotyping, the storage of that particular data is again big challenge. So the people, they are using cloud servers and other tools to store the data, but again it is a challenge, and high cost and infrastructure which is required, that is again a challenge. Another important consideration is that the regulatory framework for these gene genome editing crops, it vary from country to country, and there are concerns related to that, whether the it's not always that once you have developed the improved genotype, so it will be accepted by the public or the consumers. So it's not that always. So public acceptance is again issue, because there are concerned, the people, they are worried about whether this edited gene is useful or is harm them or is not harmful, like what we can say transgenic crops. Again the where we need to see whether this modification in gene, what we have done through genome editing, no doubt we have not what we can say use transgene, we have modified the gene or DNA sequences of a particular gene only, own gene only. But there are again concerned that when we have modified that particular gene, So the product of that gene is whether that is harmful to some beneficial insect or not. So we need to check. Again, there are issues of intellectual property. And these technologies, these are very advanced technologies, and the patents, they restrict the use of this technology by the scientists of developing countries, because intellectual property rights, they restrict the use of these frequent use of these technologies by the poor countries. So there are also differences in global policies. In some countries, genome-edited crops, they are considered like GMOs, and in other countries, they have different policy framework for genome-edited crops and genetically modified organisms. So that is again the point of concern in this case. So the future perspective, in this case, in the future, the advanced technologies, they use the AI tools to design the crops. Means the AI tools, they help in identifying the genes, they help in designing the guide RNA, they help in the prediction of that particular trait, or they predict the ideal, what we can say, genotype, or we can say new idiotype for a particular crop. So AI tools, they assist us to frame ideal and idiotype of a crop which can tolerate different stresses, as well as which is highly yielding, and which is nutritionally also rich. So AI tools, they help us in predicting the genotypic potential of different crops with the help of the data which we have collected. And then another is multiplex genome editing, means simultaneous editing of many genes. So it is again gaining momentum. This multiplex genome editing is gaining momentum because we need to modify many genes at a time to develop crops which can tolerate multiple stresses in one growing season. And another is the autonomous phenotyping. It has great means the autonomous phenotyping helps in fast collection of data, accurate collection of data, and without much of the human intervention. So this will help in reducing the what we can breeding cycle of the reducing the time in recording the observations and analyzing and interpreting the results. So digital agriculture means in future we use this noble technologies like genome editing, high throughput phenotyping, with the in combination with AI tools, to make it or to transform plant breeding from a reactive breeding to a predictive breeding or data driven breeding. And it will help in developing the crops which are what we can say more resilient to the various stresses and high yielding, and it ensures the global food security by producing higher yields under various or variable environments. So I am concluding my lecture here. So what are the main points which we have discussed or learned during this lecture is one, that the first and foremost point is that the climate change is not a future prediction. It is happening. It is affecting our crop, and it threatens the sustainable crop production. It affects the sustainability. And the second important point is that the innovative technologies like genome editing, it has helped in improving the crops in at faster pace and with more precision and with more sustainable way. The role of high throughput phenotyping is equally important, because if we could able to find get the genome edited variants, but we could not able to identify the desirable variants, then that is of no use. So high throughput phenotyping techniques, they also play role in developing improved varieties, climate resilient varieties, by rapid evaluation or by rapid collection of data and interpreting that data for the selection of the desirable plants. The AI tools, they help in translating information into results and decisions, and they try to simulate the plant responses with the variable environment, and help in getting the more precise genetic information, as well as help in taking more what we can say correct decisions as compared to the conventional methods. So it is not the single technology which can help in get but we can say facing this climate change, but to develop climate resilient varieties, we have to use multiple technologies, or we need to integrate different technologies, so that we can develop the improved varieties in sustainable way, and reduce the use of various agrochemicals to manage the diseases or pests, and we could get a healthy environment as well as the good health and nutritional rich crops. So thank you. So I would like to finish by this quote, that the in modern climate resilient agriculture, so we start with the genetic codes, bioinformatic codes. We need bioinformatic information to know the gene sequences. Then we modify those sequences, and again we need to know the machine learning codes to get that data interpreted, or the results interpreted in response, or the responses of the plants. We know the responses of the plants through the machine learning code. So thank you.
Dear participants, if you have any doubts and queries related to this topic, please can ask ma'am directly.
Hello, ma'am.
Hello.
Someone has doubt why HTP is critical for crop improvement.
See, crop improvement, it plays role in the phenotyping. So phenotyping is very, very important, because
Hmm.
when we do selection, है ना? So phenotype means the appearance of the plant. Okay, so when we use or the breeder do selection of the plants in the field, so it is not that much accurate. Means when you have to do selection from 10,000 plants or 20,000 plants or 1 million plants, है ना? When we developed, when we do crossing, or when we do hybridization, we develop segregating populations, or when we create mutations, so huge population is there. So sometimes what happened, human error, we miss the desirable plants. A large number of plants are there, we cannot see each and every plant, and second thing is, okay, if we want to record observation on each and every plant, it took lot of time, you know so means we can do phenotyping by manually also, but it took lot of time, and we do not have time, you know so if we keep on recording observation, or taking observation recording data for months on a one experiment only, so when we do the other activities, so that's why these high throughput phenotyping, that helps in what we can say recording observation, means we need not to be there in the field, and they keep on recording observations. And many times the these imaging techniques, like they sense the changes in the plant, and when we sense the change, when we see the difference in the plant, means the plant wilting is there, okay, we find, oh, stress is there, water is deficient, okay. But through high-put phenotyping, what happened, they try to sense that the plant, in the plant water is less, because sensors are there, the images, they capture images, photosynthetic efficiency is reducing in the plant, so that is, or nutrient deficiency is there, so that thing is captured through the machines or tools, and they keep on taking up images or collecting data or sensing variations in the plant day and night, you know so means they sense the things, or they take the observations before we could sense.
Thank you, ma'am. Next question is there. How to improve the global agriculture technology?
So global agriculture, there are many technologies. Means if we can say climate resilient agriculture, if the this training program is on, so means we need to use multiple technologies, multiple techniques. We can say integrated technologies. You might have heard that the different scientists, they use different techniques. So if we could able to integrate different technologies, like see, genome sequencing we did, we came to know the information of the sequences of the genome, okay. Then the functional means the scientists, they assign the functions to those sequences, means we try to find out the functions of different sequences, what sequence control what trait in the plants, so again we get that information, okay, this sequence, it is affecting this trait, so that sequence, if we want to improve that particular trait, we want to increase the concentration of a particular chemical or particular expression of a particular trait, or we do not want that particular trait, so what we can do, that information is there, we collect the information, we modify that particular gene, okay, and we try to develop a new plant type, okay. Then comes the selection. So for selection also, this high throughput phenotyping is there, we use genomic selection also, but genomic selection is okay, we do selection at genotypic level, but that is for only those what we can say loci or genes for which markers are available, so we do not means. And second important thing is that we want to do phenotyping under real situation in the field, not in the lab, okay. Whatever selection we have done in the lab, we want to see in the real field. So these high throughput phenotyping techniques, what they are doing, they are taking observation in the field only, so in the real situation, real field situation, so that data is more accurate as compared to if we are taking observation in the but we can, so if we are doing the genomic selection, so okay, we know that we did genomic selection, that is for particular trait, but about the other traits, so in high throughput phenotyping, there is a comprehensive phenotyping of the plant. So I am not saying that we are totally rely on these because these technologies are still in developing phase, means they are and everyone is not using, but in future there will be more common, because we are using more now, we are using more AI tools, and we are using more sensors, we are using more what we can say these thermal imaging, we are means we are going more digital, so we have to integrate different technologies into a one piece, so we have to use different technologies to develop a one product, okay. We also need information if we want to develop a variety which is nutritionally rich also, so we also want to we should also have information that the traits, these nutrition means vitamin A content, or vitamin C or some other nutrient content. What type of genes are responsible for that? There are different pathways, there are different techniques, genomics, transcriptomics, and then metabolomics. So means, what is our aim and how we are going to achieve it, what technologies we have, number of technologies, so we have to see which one fit best in our case, so we have to use those.
Thank you, ma'am. Next question is, why is CRISPR-Cas9 the most commonly used genome editing tool?
Because this is more what we can say flexible or easy to use. And in this case, what we why this is more commonly used, because what happened in this case, that if we want to modify different genes, okay, so earlier this zinc finger nucleases, or what we can say transcription activator like effector nucleases, we call them as talents, these are used. But in this case, in CRISPR-Cas system, what happened, if we want to modify different genes, so we only need to change the guide RNA, okay. And if we modify the sequence of guide RNA, it's okay according to the target gene, and we can use same Cas9 and we can modify the genes, means it will cut the create the breaks in the DNA, and the mutation can happen.
Thank you, ma'am. Next question is from Dr. Alok. By using AI, is it possible to predict whether a plant variety is resistant to a particular disease at the seedling stage? If so, what types of data or techniques are typically required to make such prediction?
At seedling stage, okay, so it will depend. Means some diseases they occur at seedling stage. Some diseases they occur means the development of disease also vary with the passage of time. And AI tools, yes, we can use what we can say. We can get the information about the genome sequence of that particular crop which we are using at the seedling stage. And if we have that information, then with the help of these tools we can find out what kind of sequences are there, and whether the sequences which are responsible for the resistance of a particular disease is there or not. So it helps in AI tools, they help in identification of these candidate genes, so we can find out.
Thank you, ma'am. If you have time, someone is asking to again, please explain CRISPR-Cas9 mechanism.
Okay, so we need to open the slide now. I am already sharing, means it is quite complex in the figure. Here it is. So means what happened, we in CRISPR-Cas system, what we do, we try to find the first we should have information of the target sequence, है ना? That which sequence we want to modify. So it is again a full job, means first we have to identify the target sequence for a particular character which we want to modify, okay. So then what we do, we try to develop this guide RNA, these sequences of the guide RNA, they are just adjacent to what we can say PAM site, that is protospacer adjacent site. So these sequences of this guide RNA, they are complementary to the sequences which are present to the adjacent of that particular sequence, target DNA sequence which we want to modify. So it helps this guide RNA. It helps in Cas9 protein to identify the target sequence. Okay. So when we introduce this into the cell, CRISPR-Cas means it is a small guided RNA plus Cas9 protein. Okay, so when we introduce it into the cell, so what happened? This guide RNA means what it will do? It will the complementary sequences. What happens? It will bind. There means it identify the site where the targeted DNA sequence is present on. But it will do, the Cas9 come to know that okay, the guide RNA will tell the Cas9 that here is the particular gene, target gene sequences are present. So Cas9 will, what it will do, it will make a cut there. Okay, and it will create breaks in the DNA. So you might have studied that when the breaks will be there in the DNA strand, so the in natural DNA repair mechanism in the cell, it gets activated. And then what happened, that the sometimes what happened when DNA repair occurs, so it stitches the cut ends. So when the cut ends they are joined, so at that time there will be chances of insertion or deletion of nucleotides. Okay, so when there will be any addition of nucleotide or deletion of nucleotide, so what happened, that sequence will change, ATGC, so there will be AAGC, like that. So when the sequence will change, so what happened, that when transcription happens in the that particular DNA, and then translation occurs, so the code will change, then the this amino acids will change, protein will change, है ना? So that the function of that genes will get modified. Okay, and in homology directed repair, what happened, that the one DNA sequence known, one DNA with known sequences is also put into the cell, and whenever the breaks happen, that the sequence get inserted into the that where the DNA strands broken. So that DNA get inserted between the two broken strands, and again it will get repaired, means it gets incorporated between the nicks, the two stand breaks. We call it as homology directed repair. And again, when the different sequences are got inserted within the that particular what we can say gene sequence, again its function will get disrupted, and the product will change.
On behalf of Edu-Mentors Pvt Limited and the Edu-Mentors Society for Sustainable Agriculture and Rural Development, we extend our heartfelt gratitude to Dr. Anju Pathania, Associate Professor and Head Faculty of Agriculture Sciences, DAV University, Jalandhar, for delivering an exceptional lecture on integrating genome editing and high throughput phenotyping for climate resilient crops. Ma'am, your insight presentation on the integration of advanced genome editing technologies and high throughput phenotyping has provided our participants with a deeper understanding of modern approaches to developing climate resilient crop varieties. Your expertise highlighted how these cutting edge technologies can accelerate crop improvement, enhance resilience to climate change, and contribute to sustainable agriculture development. This session was highly informative and inspired students, researchers, faculty members, and professionals to explore innovative research and technological advancement in plant breeding and agriculture biotechnology. We sincerely appreciate your valuable time, dedication, and willingness to share your knowledge with all the participants for our 21-day International Summer School Training Program on Agriculture and Allied Sciences. Once again, thank you, ma'am, for your understanding, contribution, and for making today's session a truly enriching learning experience. And we look forward to welcoming you again to our future academic programs and wish you continued success in your teaching, research, and scientific endeavors. Thank you, ma'am.
Okay, thank you. I would also like to thank Adjumentors for giving me this opportunity to deliver lecture to the students. So I am happy to share. Thank you.
Thank you so much, ma'am. Participants, you may leave now.